but let's talk about the effects of bariatric surgery. I'm going to run through the overview again, the GI tract as an endocrine organ, bariatric surgery and nutrient fluxes and hormone secretion, and then the focus on GLP-1. Again, the first hormones that were ever discovered were in the gut, and incretins were defined just after the discovery of insulin. So it's an old term, the notion that peptides or substances isolated from the gut can stimulate insulin secretion. So again, to review the anatomy of the GI endocrine system, this should be the anatomy of the GI endocrine system, the fourth slide. On the top left, you can see villi with white cells along there representing enterocytes and dark cells representing enteroendocrine cells. And there's a lot of enteroendocrine cells in the gut. There are more hormone cells in the gut than any other tissue in the body. In the bottom left, you can see an actual photomicrograph. Now the tip of the villus with the enterocytes in purple, next slide, and then circled here you see two red endocrine cells. And if you advance the slides again, you can see this schematic, which has an enteroendocrine cell in the middle, EE cell, surrounded by two normal gut enterocytes. And these are normal endocrine cells, so they have a tip that interacts with luminal stuff like nutrients, bile acids, bacteria, and bacterial products. At the bottom, you see secretory granules at the base, and when these cells are triggered, they release their secretory granules into the submucosa where it can start to signal. Next slide. This is an actual histogram, and these on the left, you can see a cut right through the tops of villi, as if you were cutting off the tips. And when you look down there, you can see stained in green number of cells for GLP-1. Again, they're scattered throughout the mucosa. Not common, but again, you have to multiply this by millions of villi and then feet of small bowel. Next slide. So again, the enteroendocrine cells have specific responses to specific nutrients. Here in this depiction, you see the two green enteroendocrine cells, and they respond to things like glucose, fatty acid, and amino acids, basic nutrients, other digestive products, short-chain fatty acids, say from gut bacteria, and these things will trigger the release of gut hormones that are collected at the base of the villi into the blood where they can float out and do their thing. Next slide, please, or next click. Now, the question is, how do these nutrients actually trigger gut hormone cells? For things like sugars, SGLT1, a glucose transporter, moves glucose into the cell, and this depolarizes it and starts hormone release. There are a number of other receptors you can see here in the light blue, calcium sensing receptors, some fatty acid sensing receptors. All these things reflect to the enteroendocrine cell what's in the gut and trigger a response. Next slide. Again, another schematic points out something else. If you look at the bottom where CCK and GLP are being released from this enteroendocrine cell, you can see not blood vessels for these things to be collected in, but actually neurons. And it's becoming increasingly clear that there are neurons right close to the enteroendocrine cell that express receptors for the hormones and can start a reflex loop, so that a lot of the GI endocrine system starts to have features of a neural circuit. And I think this is going to be really important in our understanding of how gut hormones regulate, in particular, metabolism. And so on the next slide, you can see a photomicrograph on the left, where stained in the middle of these villi, you can see these neurons that run right up through the villi. In the bigger picture, you can see a GLP-1 cell in green just sitting right close to these neurons. And GLP-1 can be secreted into the submucosa and activate these things. On the right panel, you see gut hormone cells, here stained for PYY, And you can see that these PYY cells actually interact with neurons right at their base. And this forms, again, almost a synapse. This is depicted in the schematic below, where the green enteroendocrine cell has this extension called a neuropod. And neurons interact with this neuropod where signaling can occur. So this gives two ways for gut hormones to signal, through the blood and through neural reflexes. Next slide. So gut hormones are distributed in the gut in a specific geography, and you can see here in this depiction, starting with the stomach at the top, the small intestine, the terminal ileum, etc. All these portions of the gut have specific functions. The stomach to secrete acid, the duodenum, the jejunum to absorb nutrients, and they all have principal gut hormones. Advance. So you can see that the principal gut hormones of the stomach are ghrelin and gastrin. Gastrin to stimulate acid secretion, ghrelin to regulate appetite. In the small intestine, there's GIP, which is an incretin that stimulates insulin secretion. In the ileum, GLP-1 and PYY. GLP-1 that regulates glucose, PYY, and probably GLP-1 regulate satiety. Now, these aren't hard and fast designations. For example, advanced slide, you can see that GLP-1 is also made in the jejunum. So rather than be specifically confined to various regions in the gut, it's just a trend of distribution where things like CCK are high and GIP are in the upper intestine and GLP more in the lower intestine. Advanced slide. Now let's get to the issue of bariatric surgery. Obviously, obesity is a huge problem and seems to be increasing to now the point where a third of U.S. citizens are considered obese, and lots of people throughout the world are obese. There are no good therapeutics at present. While small percentages of the population will respond to lifestyle, it's rarely long-lived, and only something like 10% of people have meaningful weight loss over time. The costs of obesity are enormous, and $40 million is a rough estimate. Probably you factor in indirect costs, and it's triple that. So, as a result, obesity surgery has become a big deal. This was initially started in the 70s and was a kind of a small part of general surgery until the 90s. And there's been a rapid expansion over the last 10 or 15 years with a tenfold, probably close to 20-fold increase in the number of procedures. Not only are there more surgeries being done, but different procedures. There's a lot of training so that there's more bariatric surgeons. And physicians and third-party payers now start to accept obesity surgery as not just experimental, but in fact good medical therapy. The outcomes of surgery have driven a lot of research in this field, so that this is one of the major clinical research endeavors now in metabolism. Advanced slide. What I'm going to focus on now are two weight loss surgeries. There are a range of these, and many of you are familiar with the gastric band and other forms of restrictive surgery. Those surgeries are worked by decreasing the amount of food that somebody can take in at a time. They don't have big effects on gut hormones, and for that reason, we're not going to focus on them any further. The two procedures that are depicted here are the gastric bypass and the vertical sleeve gastrectomy. Let's take the gastric bypass first. You can see here that the stomach is made into a small pouch. The jejunum is separated at its top and connected to the gastric pouch. Now food goes from the esophagus to this small gastric pouch and right into the mid-jejunum. The biliary and pancreatic secretions continue to flow into the duodenum, and that's hooked down below to the lower part of the jejunum. So what you have is nutrient passage through most of the jejunum before it encounters digestive enzymes from the pancreas and bile from the liver, so that digestion really takes place only in this common portion below. There's components of restriction in this because of this small pouch. People have called it malabsorptive, although it's hard to measure that. If you take the vertical sleeve gastrectomy, a much simpler procedure, 80% of the stomach is removed, and the stomach is formed into this small tube, but the intestine isn't operated on at all. The pylorus is intact, and the flow of nutrients is the natural way. It's just that by removing the stomach, metabolic changes are introduced. So the gastric bypass remains the most common procedure in the U.S., but the vertical sleeve gastrectomy is the one that's increasing in popularity. It was 5% of the procedures 10 years ago. It's now almost 30%. Next slide. Why are these procedures being done? Well, it turns out they work. This slide shows on the left weight loss or change in BMI in people with VSG and gastric band. You can see those are the two lines at the bottom. People with medical or lifestyle interventions at the top, they look like very small changes, and even though these people actually had real weight loss. But the surgical effects are much, much larger, advanced. And these are short-term results. But if you look at the right panel, advanced, these are long-term results. This is the Swedish obese subjects study that shows the effects of gastric bypass in green over 15 years. So the weight loss is large, and for most people it holds. The medically treated or non-surgical patients are blue. So that you can see the effect of gastric bypass is long-lived, and that VSG gives you, at least in the short term, comparable results. Advance, please. So what about the effects of bariatric surgery on glucose tolerance and diabetes? These results are also pretty dramatic. Advance, please. So this shows glycated hemoglobin in obese diabetic subjects randomized to medical therapy, sleeve gastrectomy, or gastric bypass. You can see that all improved in the first 6 months of the study, but the diabetic control in the medically treated people gradually waned. This is what we see with most treatments for type 2 diabetes. They work for a while, and then the effect wears off. The surgical effects are bigger, and they're more long-lived. Here, out to 3 years, still with a nearly 3% drop in A1C. If you look at it a different way, how many medications were the subjects taken, you can see that the people on medical therapy actually have to increase their therapy, while the people that had surgery used much less medication. And again, this has really been the eye-opener, is just what a dramatic effect bariatric surgery has on diabetes. Next slide, please. You can see that within 2 years, there's a 72% remission rate of diabetes with Roux and Y compared to 21% with medicine. And this holds over 10 years, although in both groups, there's a progression. And the effect of bariatric surgery on diabetes is not permanent, although it is fairly long-lived. Advanced slide, please. What has caught everybody's attention is the rapidity of the effect of surgery on diabetes. Certainly weight loss and improvement in insulin resistance is a major factor in how people handle glucose after surgery. But it's been long noted by surgeons that the diabetes was getting better even before there was a lot of weight loss. And you can see that in the left panel, that the fasting blood sugars in this cohort of diabetic patients were going from nearly 10 millimolar to 7 millimolar within 6 days. And in 6 days, there's a little bit of weight loss, but not a lot. If you look at insulin resistance, that improves dramatically in 6 days. So that these observations led people to say, boy, surgery's doing more than just causing weight loss to improve diabetic control. And that really set off a lot of research, and then the focus on gastrointestinal hormones. Advanced slide. So let's talk about the effects of surgery on GI hormone secretion. And we'll focus on GLP-1 because that's where the effects are most stark. So GLP-1 is produced in a specific enteroendocrine cell called the L cell. And L cells make GLP-1, they make other hormones, they make GLP-2 and oxyntomodulin. All those come from the same prohormone, proglucagon. L cells also make PYY. Something about bariatric surgery causes L cells to secrete more hormones. And you can see that on this three-paneled figure with oxyntomodulin at the top, GLP-1 in the middle, PYY at the bottom. If you look at the control subjects, the non-operated subjects, you barely see a change in any of these hormones at all. And that's because the y-axis is so skewed by the humongous response in the surgical patients. So they have sort of a 5, 10, sometimes even 20-fold greater GLP-1 response than people that don't have surgery. And all the other L-cell products also come out in abundance. Advanced slide. This effect is not transient. These are GLP-1s in patients who had surgery and were studied compared to controls at 9 to 15 months, at 21 to 30 months, and at greater than 36 months. And in fact, there's lots of studies now that show that the hypersecretion of GLP-1 after gastric bypass surgery persists for years. Advanced slide, please. This slide just shows that on the left, GLP-1 goes up a lot after gastric bypass, within a week and persisting for 3 months, but it also goes up a lot after the VSG. So that both of these procedures, although they're very different anatomically, the gastric bypass relatively complex, the VSG relatively simple, they both have similar responses on GLP-1. The concordance here, because they also have similar responses on diabetes control, has raised the question as to, does GLP-1 account for this effect? Next slide, please. So why does bariatric surgery increase GLP-1 secretion? This schematic gives us a little bit of insight. You can see on the left the anatomy of a gastric bypass, and on the right the anatomy of a VSG. In both cases, you have rapid passage of nutrients from the mouth, down the esophagus, and into the gut. In the VSG, it's because the jejunum is hooked right to the stomach, or in the gastric bypass. In the VSG, it turns out that tube really speeds the delivery of nutrients. Things don't sit around in the stomach, but they pass right through. And so in both of these, you have rapid passage of nutrients from the bottom of the esophagus to the well down the jejunum. And it's thought that this sort of abrupt surge of nutrients is what triggers GLP-1 secretion. Advance, please. Now, these are data from an old study, but they contrast the two incretins. And if you look at the left panel, you can see GLP-1 on the left and GIP on the right. And these are subjects that drank glucose, either 50 grams in the upper panel and 100 grams in the lower panel. And what you notice is after they drink glucose, the GLP-1 secretion goes up in a spike and then comes down. In contrast, GIP goes up and stays up as long as there's glucose in the gut. And the notion here is that part of the GLP-1 response is not just having glucose in the gut, but having it get to the gut rapidly. This early spike in GLP-1 secretion during this oral glucose tolerance test happens at a point where glucose is flowing rapidly from the stomach into the intestine. If you look at the right panel, you see this even in a more simpler form. These are subjects that had their duodenum cannulated with a tube, and then they were infused with glucose. And you can see that the GLP-1 response only goes up where they give a rapid rate of nutrients and not when they give a slow rate of nutrients. On the other hand, when you infuse glucose into the gut, GIP comes out in a dose-dependent effect, but it's secreted in either case. So there's something about rapid passage of nutrients, glucose and other stimuli, that makes the L cell particularly likely to respond. This may be a neural connection from the upper gut that triggers a reflex to L cells, or it could simply be that when there's a lot of glucose, more of it flows downstream where there's a denser population of L cells. These are questions that are still being asked, but I think the one conclusion is clear. More rapid passage of carbohydrate, glucose, other nutrients into the intestine gives you a bigger GLP-1 response. Advanced slide, please. So if we were going to summarize the effects of bariatric surgery on gut hormones, this is the data that, as it stands now. L-cell products, GLP-1, PYY, and GLP-2 are increased, sometimes a lot. GIP, which is an upper gut hormone, and is actually, a lot of the GIP cells are bypassed with a Roux-en-Y gastric bypass, don't really change very much after bariatric surgery. Ghrelin levels, again, a stomach hormone primarily, are decreased. And some people, so the surgeons always say it's because I cut out all the ghrelin cells. That may or may not be the case. What argues against that is ghrelin isn't suppressed in every patient with surgery, but in general, ghrelin, which stimulates appetite, goes down. CCK, which is a hormone that's involved in gallbladder function and pancreatic enzyme function, also has an effect as a satiety hormone, and CCK levels tend to be high. Gastrin, again, bypassed in both of these procedures, doesn't change very much. There's enough gastrin cells remaining that you can still measure gastrin, but it doesn't go up. And then other gut hormones just haven't been studied. So this is really the status of gut hormone responses to bariatric surgery as we know it now. Advance slide, please. Now, the other thing we know about surgery is that it definitely changes the glucose fluxes. I mentioned that if you give somebody a glucose tolerance test, it gets into the intestine much faster after surgery. This just shows that it also gets into the blood more rapidly. You can see in the left panel plasma glucose from a group of people that had a Roux and Y, those are the squares, control subjects with no surgery, those are the open circles, and then a gastric band, those are the triangles. And the glucose rises much more quickly in the subjects with a gastric bypass. And in the band and the control, the glucose level is about the same. So there's a faster increase in blood glucose and a faster clearance as well. The levels come back to normal more quickly. And if you look on the right panel, you can see that this rapid rise in glucose is just all from the gut. That's what the top panel shows, is that's gut-derived glucose. And in the bypass, it's really dramatic. Next slide, please. This shows a subject with a gastric bypass and also a tube in the remainder of the stomach and the duodenum. This patient had a tube placed for decompression, and we used that to measure what glucose looked like when you gave it orally and through the tube. And you can see again, when this patient drank glucose, she got a big rise, a big rapid rise. That's the top left panel. When we gave glucose through the tube, that's the top middle panel, the glucose excursion was more normal because that was coming through her sort of normal anatomy. Advanced slide, please. The GLP-1 levels were much higher when she took glucose orally as well. And you can see on the top right panel just how much her glucose level dwarfed some non-operated controls. Next slide. Now, do these high GLP-1 levels mean anything? Do they actually have a physiologic effect? Well, this was studied in a group of bariatric surgery patients and control patients who had their blood glucose levels clamped and then took a meal to stimulate the incretins, and insulin secretion was measured. And then in a second study, GLP was blocked with a GLP receptor antagonist. So these studies allow a controlled study of insulin secretion with and without GLP-1. And you can see in the two left panels the blood sugar levels in the bypass patients, and then in below the insulin levels. So the insulin levels are really high in the first study, and then when you block GLP-1, they drop a lot, maybe 50%. In the controls, in the bottom right, you can see that blocking GLP-1 really only had a minimal effect, such that if you look at the bar graph, you can see that in patients with a gastric bypass, the GLP-1 effect, or the percent suppression of insulin when you take away GLP-1, is about threefold higher in a person with a bypass than a control. What these data tell us is that bypass patients who have these really high GLP-1 levels actually respond dramatically to GLP-1, and it's one of the things that drives their insulin secretion. Advanced slide, please. So let's talk as a more sort of illustrative example of the syndrome of hyperinsulinemic hypoglycemia after bypass surgery. And this is a syndrome that's becoming increasingly recognized. It's typically described in people who are a year or two after bypass surgery. Usually they've had substantial weight loss. Almost never were they diabetic before surgery, so usually they had normal glucose tolerance before surgery. And what they start describing are typical symptoms of hypoglycemia happening an hour or 2 hours after they eat, with adrenergic symptoms of sweatiness, shakiness, tachycardia, hunger, sometimes becoming severe enough to give neuroglycopenia with loss of consciousness or seizure. The prevalence is hard to tell, and some people have said it's as little as 0.1% or 1 out of 1,000, and others said if you do a careful study, you could see some dip in postprandial glucose in up to 15% of gastric bypass patients. I see it as a continuum where most people that have gastric bypass will have some fluctuation in their blood sugar and sometimes drop it, but only a small percentage get clinical symptoms that are limiting, that is, that prevent them from driving, cause them to have problems at work, etc. Well, so the question is, what separates these patients from others? And one suggestion was, well, it's GLP responses. Next slide. And here you can see plasma GLP-1 levels in controls in the left side. In control patients, those are those little flat lines at the bottom. In bypass patients without symptoms of hypoglycemia, those are the open circles kind of in the middle there. And then in a small group, about 7 patients with hypoglycemia who have higher GLP-1 levels. Now, what I can tell you is this was the first description of this, and there's been a number of other studies where the GLP-1 levels in the symptomatic and asymptomatic bypass patients don't separate this much. This was the one paper that showed the biggest effect. On the other hand, there's no question that these people with hypoglycemia after surgery certainly have elevated GLP-1 levels. Next slide, please. Now, if you look at these patients again, you can see here islet hormones and incretins in patients that are symptomatic, bypass patients with hypoglycemia that are asymptomatic, bypass patients of the same age and relative weight who had surgery about the same time, and then non-operated controls. And again, the blood glucose after a mixed meal tolerance test is much higher in the bypass patients, that's panel A, than in the controls, that's the gray line there. If you look at the insulin, that's panel B, that's also much higher in the bypass patients and higher in the subjects that have hypoglycemia. So this is a hyperinsulinemic hypoglycemia. If you look at panel D, you see that the glucagon levels are actually higher in the bypass patients too. And this is curious. It's independent of hypoglycemia, but it's been described. The GLP-1 levels are higher in both groups of bypass patients. And here the difference between the hypoglycemic and non is not so important. And you can see here the GIP levels in this particular series were actually higher, at least at the beginning, in the bypass patients. Next slide. Now, this study took some of these patients with hypoglycemia and some of the asymptomatic controls and studied them on 2 occasions. They gave them a can of Ensure to drink once and then brought them back and gave them a can of Ensure when they blocked GLP-1. And if you look at the top left panel, you see the blood sugar in the hypoglycemic group, the dark line. They get a rapid peak of glucose, and then they bottom out. And in this study, all 10 subjects had hypoglycemic symptoms. They had autonomic hyperactivity and mild change in sensorium. But over time, they bounced back. If you look at the asymptomatic patients, their blood sugars go up fast, but none of them got hypoglycemic, and none of them were symptomatic during this study. And then in the far top right, you see the control subjects with smaller glucose excursions and no hypoglycemia. Again, the insulin responses were huge in the hypoglycemic patients. That's in the bottom left, and even bigger than the asymptomatic patients. Now, when the patients were given the GLP-1 blocker, you go back to the top left and you look at the dotted line. All 10 of the hypoglycemic patients now have a normal glucose excursion. None of them get hypoglycemic, and in this study, none were symptomatic. The GLP-1 blocker had minimal effects in the asymptomatic patients and the non-surgical controls, and you can see the dotted lines in the top middle and top right. The strong inference from this study is that if you can block GLP-1 in a bypass patient having hypoglycemia, you can fix it, such that GLP-1 seems to be pathological in this case. Now, this is a limited study. I would tell you that the subjects that were hypoglycemic were hand-selected to be the most symptomatic patients that we could find. And the asymptomatic patients were hand-picked to have no symptoms, so they were dramatic polls so that we could really test the question. But based on this study, you could say that bypass surgery stimulates GLP-1 secretion in everybody, but that a small group are either more sensitive to GLP-1 or have slightly higher GLP-1 levels and so get hypoglycemic. The problem is that there's no good GLP-1 receptor blocker that we could use to treat these patients, and we still do things like encourage them to take small, frequent meals, maybe use a little bit of acarbose to slow their absorption of carbohydrates, limit their carbohydrate intake, etc. Not very satisfying in a lot of patients. Advanced slide, please. So that the conventional model of GLP-1 action is one that fits with what I've just described, and that is GLP-1 released from the gut into the portal vein, gets through the cardiac circulation and out to the target tissues like the beta cells where it can have its effect. And I think this model fits with bariatric surgery where the GLP-1 levels are so extraordinarily high that the peptide actually isn't metabolized to such an extent that it can't have endocrine effects. Now, the next model is a model that people are studying, and that is a little more subtle. But it is the model where GLP-1 doesn't act as a hormone, but has most of its effect right at the gut, down in the bottom left of this figure where it says villus, and you can see the little black dots of GLP-1 being released by the L cell and triggering this neural reflex that's communicated to the hindbrain, shunted to the hypothalamus, and then down through the autonomic nervous system. And this is a model that has some experimental evidence supporting it and is under intense investigation. I think that in patients with gastric bypass and very high GLP-1 levels, both of these models are probably at least partially in play to explain some of the effects. Again, what's interesting to speculate about is the differences between the patients that have hypoglycemia and those that don't. Do they have actually higher GLP-1 levels? Do they have higher responses on target tissues? Or might their neural system be more sensitive to GLP-1? I think these are all interesting questions that we'll probably figure out in the next few years, but will tell us a lot about, one, mechanisms of bariatric surgery, and two, the importance of gut hormones. So last slide, to summarize, the gut is filled with hormones, and these regulate GI functions, motility, secretion of pancreatic enzymes and acid, and emptying of the gallbladder. But they also have big effects on metabolism. No question that some of these gut hormones play a role in satiety. They certainly, the incretins, GLP and GIP, are very important for insulin secretion. I think we're just starting to learn about how this gut communication and metabolism is working out. Roux and Y gastric bypass and vertical sleeve gastrectomy both increase the secretion of GLP-1, and not a little bit. We're talking about 5-fold, 10-fold increases, so dramatic, huge increases in GLP-1. The effects of these surgeries on other GI hormones now are not nearly as dramatic. The suppression of ghrelin is probably the next most compelling, but it's GLP-1 that really seems to be the hormone that responds to bariatric surgery. GLP-1 is a nice model for how surgery could act through gut hormones, and I think the example of hyperinsulinemic, hypoglycemic is a good model. That being said, I think there's a lot of data now that's emerging that GLP-1 is not the whole story in gastric bypass nor in vertical sleeve gastrectomy. Both animal models and human models have demonstrated pretty clearly that there's a lot of other things going on, and that GLP-1 probably plays a small role, maybe in these hypoglycemic patients, maybe in some aspects of insulin secretion. But things like weight loss, appetite, other GI functions, and probably the resolution of diabetes are all regulated in a much more complex way. So I'll stop there and take any questions that have come up during this presentation. Thanks very much.
Great. And thank you for a really interesting presentation. So we do have time now to address some of the questions that came through during your presentation. And I'll start with a couple of questions from one audience member. Are bile acids involved in the GLP-1 dependent processes? And if yes, how do you think? In addition, what do you think about studies from animals, rats and mice, looking at bile acids and or GLP-1?
Yeah, so those are really, really good questions. One of the things I didn't talk about was that in both VSG and gastric bypass, not only do nutrients go through the gut fast, but bile acids go through the gut fast. And the distal intestine, where bile acids are reabsorbed, sees more bile acids and sees them quicker than normal. The other thing is that both of those surgeries are associated with increased circulating bile acids, so that the whole pathway, the whole enterohepatic circulation of bile acids seems to be sort of turned up in bariatric surgery. It's pretty clear that bile acids have direct effects on L cells to augment GLP-1 secretion. And so not only the increased rates of nutrient flux, but probably increased rates of bile acid flux also contribute to the elevated GLP-1 that we see in these surgical patients. I think that the circulating bile acids beyond GLP-1 probably act in the circulation to directly mediate a variety of effects. And this is a very interesting area of research. That is, what responses of bariatric surgery are mediated directly by these increased concentrations of bile acids? And that's an open question. In animal models, if you knock out one of the bile acid receptors, you don't get a weight loss effect with an experimental bariatric surgery in mice. And the mouse studies have gotten amazingly, have an amazing ability to replicate what we see in humans. That is, people can actually do now VSGs and, to a limited extent, gastric bypasses in rats and mice. In mice, we see that knocking out the GLP receptor doesn't eliminate the effects. Those animals still lose weight, and they still have pretty good glucose tolerance. So it seems like there must be some adaptation to the loss of GLP-1 signaling. But I think that the animal studies, to get to the second part of that question, are going to show us a lot about mechanistic targets, and it's a good way to look at some of these candidate mediators of bariatric surgery.
Excellent. Well, it looks like that is all in terms of questions from the audience. So this will conclude our webinar today. I'd like to thank our distinguished speaker, Dr. D'Alessio, and also everyone who joined us today, and hope everyone has a great rest of the day.
Thank you.